So, hi everybody, I'm Daniel and I'm presenting with Faith. And we're going to be talking about ourselves and our research. And I guess one of the things that we're thinking about that links our research together is that we look at biological, I put energy, but also like material, biological energy and material flow through natural and engineered systems. So first, like I said, my name is Daniel Delgado, and after high school, I went into the Navy, where I wasn't on a submarine, so I'd be underwater for like 2 months at a time. So this isn't too bad as far as being used to social distancing, but still a little rough. I was a nuclear plant operator. That got me into engineering, and I liked it, but I didn't want to stay in something so mechanical. So when I was looking for what type of engineering to go into, I decided to go with environmental because I really liked how it had biology and chemistry in it. So right out of the Navy, I went into community college and then transferred to San Diego State University. There, I was doing some research, and well, I started doing research. I applied for a lab position and got accepted. And my professor was growing algae to extract the fats inside of the algae and use it as fuel. And he gave me this project to compare how well the algae grew, or how much fat I was getting from the algae that I was growing in the Miracle-Gro. That's what we were using to grow all the algae, versus wastewater. And I also was looking at how well the algae was removing nutrients from the wastewater. That's what really got me into wastewater. And that's when I decided I really like biological treatment, because that's what the algae were doing. They were treating the water. And then right before I finished my bachelor's degree, I was looking at PhD programs. And I was really interested in USF because a lot of their biological process research, and I really liked how they did a lot of work with developing countries, or like a lot of international work. So I applied there and got accepted, and then that's where I'm at now, doing wastewater treatment research. I'm looking at on-site wastewater treatment. So that's specifically, I'm looking at septic systems to remove nitrogen. And this is one of the wastewater treatment plants that I work with. I get my wastewater treatment samples, or my wastewater, from them. We used to have a habit system there, but it has since shut down, but that's still part of my research. And with that, I'm going to switch over to Faith. I'll stop sharing.
Hey guys, my name's Faith. Oh.
But we still,
Oh wait. I think you keep sharing the screen, and then it'll go to like your research after.
Okay, that works.
Sorry about that.
Sorry, we're gonna switch a little later so we can show our research a little bit better. But go ahead.
All right, so hey guys, my name is Faith Frings. I'm currently an undergraduate student at USF. I gained my bachelor's degree in marine biology, and I'm graduating this semester. I chose the marine bio path when I was in high school. I did a lot of scuba diving, and I got a lot of experience kind of working in the ocean, and I fell in love with it, and I wanted to learn more about marine conservation. I wasn't sure what I was going to do with it yet, but I knew I wanted to work in marine biology. And so I came to USF, and I did some tutoring in some of the bio and chem classes, and I really fell in love with teaching. And so I decided to sign up for being a learning assistant, which pretty much is I did for bio too. A learning assistant is where you kind of help out the professor teaching the class. So it's more of a flipped classroom setting where the professor will teach for about 15 minutes. They'll then give all the students a worksheet to do. And then as a learning assistant, you'll help about a group of 10 students kind of like go through the worksheet, make sure they understand all the principles. I really like doing that. And so I wanted to like kind of learn more about pedagogy and bioeducation. So I volunteered in Dr. Provost's lab. She is a professor at USF, and her lab kind of focuses on bioeducation and kind of understanding students' thought processes as they look at different biology problems. So I started volunteering in her lab in November of last year, and I really like fell in love with teaching and kind of like understanding more about her research. And so I decided to apply for the master's program at USF to get my master's in ecology and evolution. Thankfully, I was accepted, and I'm going to be a master's student next semester in the fall. I wanted to get my master's in ecology and evolution because I'm kind of interested in how students understand phylogenetic trees, as well as how they understand different biological principles such as like energy flow in an ecosystem. After my master's, I plan on getting my PhD in bioeducation and hopefully teaching biology at a collegiate level. And that's pretty much me.
So a little bit about my research. Like I was already talking about it, saying that I look at on-site wastewater treatment systems. So this is one of the islands that I work with a group of engineers and anthropologists, and it's called Strong Coast, and we're like focusing on food, energy, water systems. So this island is a perfect example because it's a tiny island and self-contained. So the water that we get from here, the people come into the island. This is a national park that's just off of a coral restoration site. So they're growing these corals to try and replant, like cover the area that used to have healthy coral with healthy coral. But they're having this problem where because of the island, they're getting a lot of, well, it's not because of the island. That's actually one of the things that as working with anthropologists, you kind of learn about the people part of the of the systems that you're working with. So there's this island. They have people coming here. They use these bathrooms, and it is very likely that the extra nutrients in the water that are causing these algae growths that cover the coral, that affect the coral's ability to grow, is and eventually kill them off, is caused due to the system that they have, the water treatment system that they have. So I'm looking at how to improve septic systems so they can remove more nitrogen. And that's also something interesting where like my research before, I was trying to grow algae because it was beneficial, but in this scenario, it's causing problems. So this is a simplified nitrogen cycle. This is the nitrogen species that I look at. Like I was saying, they're getting a lot of nitrogen in the water from people because there's a lot of nitrogen in our urine and feces. So so nitrogen in the air, we turn it into, well, not we can turn it into organic nitrogen. Some organisms can turn it into organic nitrogen, but we've learned to do it through industrial means. So we put a lot of stress on this side of the nitrogen cycle. So now we got a lot more organic nitrogen, and because we use it for fertilizers, we eat the food, we then like concentrate all these nutrients, and then it becomes ammonia and ammonium. And then in the systems that we use aren't doing anything to remove this, we end up with extra nutrients in the water that lead to algal blooms like they're experiencing in Belize. So my work is on how to improve, let me switch to the pointer, how to improve nitrification. That's this part right here where we turn ammonia into nitrite and then nitrate, and denitrification. So we're turning it into nitrite and then back into nitrogen to leave the water, and then it's now in the air, no longer a contaminant in the water. And in the air, it's inert, so it's like most of the air that we breathe. So what I'm looking at to improve the system that they have there, this is what it looks like in the lab. And this is what it looks like as a schematic. I'm looking at how to improve this side, nitrification, and this side, denitrification. So like a normal septic system, I have it set up to get wastewater from the water treatment plant that I was talking about through a pump on a timer. That's this thing right here. So that way it can mimic the type of flow that septic systems see in the home. And as water comes in, it flows through gravity from the septic tank right here. into the nitrification filter. Here I'm using bacteria to turn the organic nitrogen into nitrate nitrite. This is the second phase of my experiment. I'm not there yet. So what I'm trying to find out is what's happening inside of these, I've been calling them reactors, so I'll stick with that. What's happening inside of these reactors. And I guess I wanted to ask you guys if you guys have any idea how I could do that. How can I find out what's going on inside of here? And I guess, are we doing Q&A right now, or can we ask questions and have them type it in?
You could certainly ask questions and have them answer, unmute themselves and answer.
Okay. I'm sorry, I don't know how to do that. But yeah, if you guys can.
Oh yeah, they can like unmute themselves. So you ask a question and then they can unmute themselves and then answer your question.
Any takers? No?
So how might Daniel know what the microbes are doing in his biofilter? He's got wastewater going in, and he's got something called effluent or treated water coming out. He wants to know what's going on inside. What do you think he should do?
He could put in a sensor. You could put a sensor that senses the nitrogen levels before and after the reaction. I don't know. I'm just taking a wild guess.
No, that would be great. Unfortunately, those kinds of sensors are very expensive. But at the wastewater treatment plant, they have something called a chem scan system, and they can use different kinds of probes and sensors to measure the concentrations in the wastewater. Any other thoughts about how you might do this?
Okay. Well, the way I do it is I see what's going in and what's coming out, and then we make some assumptions that the amount of stuff inside here is not really changing. There is some biomass growth, but that's microorganisms as they eat and they get bigger and they multiply. But overall, there's no accumulation in here. So if I take in minus out, I can see some of the water qualities. And based on what I have inside of the water, I can kind of understand what kind of reactions or what kind of processes are happening inside of here. So the way I do that is this septic tank right here that leads to this, my nitrification column. I'll take what's coming out of the septic tank. We call that effluent, but it's the second reactor, the nitrification filter. That's its influent. That's what's going in. So it's influent for the nitrification filter minus effluent. And so I'm going to get a sample from here. I'm going to get a sample from here. And then I get something like this. So this is from Habits. This is the other system that I was talking about, where the system that I'm working on now has a lot of salt water in it, but this one was fresh water. But they're very similar. They're both trying to remove nitrogen. So I have like pH, dissolved oxygen, alkalinity, a whole bunch of nitrogen species, and like tint, for example. This is one of the ones that I'm very interested in. Total inorganic nitrogen. So that's the nitrogens that I was talking about earlier, the ammonia, the nitrate, nitrite. And I have I'm sorry, let me get back to that one. So I have what's going in, that's the influent right here. And I have what's coming out, that's the effluent. And if I plot that on a graph over time, I get something like this. So let's say on day 20, this is what's going in and this is what's coming out. There's two different reactors. So we were testing out one system that had water going in recirculation and water that was just went straight to the system one time. So recirculation one is going to perform better for several reasons, but that's these two systems right here. This is what's coming out of one of the reactors. So this would be like my nitrification filter, and this is what's going in. So like over time, I can see a trend of how well it's performing its job. And if I average those out, I can get something like this, where I can get an overall performance of the system, where I have the influent minus the effluent. But these figures are more for papers. Like something like this is a little bit more exciting for a presentation, and it also tells you a lot more about what's going on. So whereas the last figure, I only had total inorganic nitrogen, this time I have different nitrogen species, so I can kind of get a better idea of what transformations are going on. Like you can see here that some of the organic nitrogen, a lot of the organic nitrogen is gone, and now I have all this gray stuff, which is nitrate. So I know I'm having nitrification occurring from here to here. And that's kind of how I do the research that I'm working on now. That I can switch over to Faith. Stop sharing.
So I'll share my screen. Here we go. Yes. Go as fast as possible. All right. All right. Just thinking about it. Okay. All right. Hey guys. So thank you, Daniel, for giving us insight in your research. Mine's a little bit different. So my research kind of more focuses on using a computer. So we don't get to do a lot of like cool field stuff, but we do get to kind of it's more teaching. So it's kind of like a pedagogy research. So what we're looking at is we're kind of looking at a food web and kind of how students understand the food web at an undergraduate level. So first I want to give some kind of background information, as I'm sure you know. So this is an example of a food web, which is a visual representation of how energy and matter flow throughout an ecosystem. So I have two examples of food webs kind of show that they can look very different in different ecosystems. Like for example, your primary producers in a terrestrial ecosystem might be like a plant or a mango. However, if you're in an Arctic system that is mostly marine, keep touching, you're going to see that your primary producer is going to be algae. And so this kind of shows that food webs kind of like can look very different based off where you're looking at them. And so my research kind of want to look more at how students understand these food webs and misconceptions they might have. So my research project was mostly stemmed from a larger research project from a PhD student in the lab, where he showed students this web. As you can see right here, this is an example of a food web. And he would ask them questions about the food web and kind of looked at their responses. So I'm going to ask you guys some of those questions. No pressure, but just kind of like trying to check it out. So one of the questions he asked was, if E is decreasing, what is going to happen to P? What do you guys think?
P will decrease.
Exactly, yes. So in this food web, since the energy, this is kind of like your primary producer right here, since E is giving energy to P is the predator. If there's less E, then there's going to be less P. And another question that he asked is a little more complex, but you guys seem very smart. If E decreases, what do you think is going to happen to R?
I'm pretty sure R would increase.
Increase? Why do you think R is going to increase?
You said if E increases, what would happen to R, right?
Oh, no, I said if E decreases. Sorry about that. I might have said increase. That was my bad. What do you think is going to happen to R if E decreases?
I think that R will decrease because since E's not going to be there, it has to find another food source, which is L. And then after L gets eaten by R. Like, I don't know.
Yeah. So that's one way of looking at it. So there's not really like a wrong answer. So we pretty much made that's a good. Thank you so much, Sammy. So what we kind of looked at is that we hypothesized that students would say that if E decreases, then P is gonna switch over to eating more R. That would cause R to decrease. However, in our when we did it, we analyzed the results. We saw students would actually take what we call the long path, is where they would actually go this route, say that L would have more food since it's not competing with E, and that would cause R to increase, which is another way of looking at the problem. So thank you for your response. That was kind of like the other way of looking at it that we saw. That was kind of different from our initial hypothesis. So throughout the coding process, while we were looking at this the questions, the answers, we saw that students would often reverse the arrows and when they were responding. So they would say that E is actually eating P. And so we thought that was a common misconception that was happening. Another thing that we saw that was very interesting was that when students would talk about competition and the responses, even though they weren't prompted to do so. So they would say that L and E were competing with each other, or R and E were competing with each other. So what my research project then was kind of looking at the correlation between students reversing the arrows in their responses and talking about competition. So there were two types of competition that I was looking at. One was interspecies competition, the other was intraspecies competition. Now, can anyone tell me what interspecies competition is?
Is it like competition between one species?
So very close. So that's intraspecies competition. Interspecies is in between two species. So it's a very common like concept that when I was learning biology too, I would often mix them up. So intra, kind of think of intra as in, like in itself. So intra would be competition within one species. For example, you could say, oh, P is competing within itself for a resource of R. And then inter would be in between two species. And I want to thank everyone who like answered my questions. Sorry for putting you guys on the spot, but I really appreciate all your feedback. And so now I'm going to kind of show what we did. So we did all of our coding through Excel. And pretty much what would happening is we would read about 20 of the responses on the left. So we would have kind of this is question 3. So they would ask the question, and then they would then explain their answer. And so what we would do is we would make a coding rubric based off about 20 of the responses that we read and kind of see what they were saying. So the larger research project is kind of in blue. I hid most of the codings that he was using, but what I did is we saw that error reversals were happening when we were coding for Chris's project. And so I used those in my research, and then I made a coding rubric kind of looking at the different types of competition that students were mentioning. So of the large sample size, we saw that 227 students mentioned competition in their responses. And so these are some of the responses that they had. And what we would do is we would read the response and then code for the different answers that we saw. So for example, in this response right here, you can say that then competition would increase. So since the student said that competition increased, they would get a 1 for increase and a 0 for decrease. And since they mentioned how there would be competition within P, we would then put a 1 for interspecies competition within P. Now there's other paths that we looked at, which would be other, which talk about competition within F, and then interspecies competition, which is kind of looking at the ERP path as before. So this is kind of the short path. They talked about interspecies, and then the long path would be kind of going through O, through L to R. So this is kind of how we did it. It's pretty much just kind of like typing in 1s and 0s, and then you can then do statistical analysis using this program called R, and R Studio. And pretty much what we were looking at is we did what was called a phi correlation. So that's kind of a type of statistical analysis where it looks at the correlation between two different variables. For example, since I was looking at the correlation between error reversals and competition, I would correlate how often the student says error reversals and whether when they did reverse arrow, they also said interspecies competition, and then when they reverse arrow, they also said intra-species competition. So I looked at those two variables, and so these are kind of the results in a graph form. And so right here, you can see, so what you want for your phi correlation is if the value is closer to 1, that means that there's a strong correlation between the two. It could be positive or negative, just you want the value closer to 1. And so what I saw is that in question 3, only 3% of the students reversed the arrows. And question 3 was asking if E decreases, what happens to P? So that was more of a direct effect question. And so I kind of drew the arrows this way to show the reversal. And of the students, of the 3% that did reverse the arrows, most of them were talking about competition between R and E for P. So majority of the students would say that if E decreases, that means that R would have less of competition with E for P, and R would increase. Now the 97 that had the correct error flow right here, we also saw promising results, and they would say that there would be more intra-species competition within P, which means that P would then decrease because there's less resources. So that's what we were looking for. For question 4, we saw that students, so this is the question that was asking if E decreases, what happens to R? So this means that we saw that students, about half of them, end up reversing the arrows in their responses, as shown here. And of those, about half, they would talk about the same thing in question 3, where they would say that R would have less competition with E for P and would end up increasing. So of the students who had the correct arrow flow, we saw, like what Sammy said earlier, how we saw a majority of the students actually talk about connecting R to E through what we call the long path. So they would talk about how L would have less competition with E for O, and then therefore R would then increase. So we saw that a lot of students had like a lot of problems and misconceptions with more complex food questions. Like question 4 was looking for a more indirect effect. And so we're kind of trying to figure out a way of fixing this problem, and we think about a good solution would be to give students more practice with formative assessment questions, such as like clicker questions, giving them more practice with food webs that not only involve organisms, but also involve like letters and numbers, like here, where it can be trickier, because if you don't know that, so a lot of students understand that, oh, the hawk is going to eat the mouse. But if you just give them a letter and a number, it's kind of hard to determine like where the energy flow is. You can kind of see what their thought process is. So that's kind of what my research was this semester. And then when I go to get my master's, I'll start a new project. And that is all I had. So thank you, guys. Are there any questions for both of us?
So Faith, what's the goal of your research? Are you trying to, I guess I'll just leave it at that. What's the goal of your research? What would be success for you?
So success for me would be seeing higher test scores in intro in biology students exams in upper level classes in college. So pretty much what we like for we're trying to figure out more efficient ways of teaching students biology in order to help increase like their knowledge of these principles and help them in upper level courses. So for example, when I was a learning assistant, we would kind of, this was like the first semester, it was kind of a new thing everyone was trying out. And we saw that between the previous years where students were taught with a traditional, lecture, and looking at the test scores of students in the learning system as like in that type of environment, we saw them have higher test scores and that they understood these more conceptual questions better. So pretty much my goal is understanding where the misconceptions are. So for example, students were flipping the arrows in their responses. So giving them more practice around that kind of concept of how the energy flows in the ecosystem, and kind of helping them better understand this concept so they can succeed in upper level courses such as like genetics or ecology.
Any questions from the students for Daniel or Faith? I had a question for Daniel, actually. You showed the bioreactor and you showed the data, but you didn't really explain where the data came from. Can you talk a little bit about how you do those measurements, since I know that's a big part of yeah.
Yeah, so like I showed where I would get the water samples. What I do from there, you're asking about how I get an actual measurement from it though, right?
Right.
So there's a lot of processes that we do. Like some of them, like pH, dissolved oxygen, and conductivity, those are all going to be, well, those we do with a probe. So we'll stick a probe into the water and it'll give us a reading. Stuff like nitrogen, we either use an ammonia analyzer. So after I get the water sample, I'll run it through a filter to remove a lot of the solids that are still in there. And that way it doesn't mess up the machine. And then I'll just get the filter is made so that pretty much only what's dissolved in the water is going to get through the filter. So then I'll have like the dissolved concentration of what I'm looking for. And that's what I'm most interested in. So I'll run it through, for me, I'm using an ammonia analyzer. And what it does is, like, once I put this water sample in there, it'll take it has a auto sampler. So it takes the machine itself takes this needle, pulls out some like a milliliter of the sample or half of a milliliter. And then first it measures the ammonia. It'll heat it up, and then actually it measures the gas, right? I wasn't entirely sure. It measures the gas after it heats it up, and then it'll look for the other nitrate and nitrite. But with that one, first it converts it to ammonia and then measures the ammonia in the same way that it measured it, just right out of the water. So then I'll have the machine is connected, or the analyzer is connected to a computer that we have a software that'll tell us what the concentration is. From there, well, actually, it doesn't just tell me what the concentration is, it'll read it as a conductivity. And the conductivity, I'll use some standards that I know what the concentration in the water is, and then it'll do a comparison of the conductivity versus the concentration of the samples that I used to kind of have it'll have like a graph, kind of like a regression line to see, like, at this concentration, I'm getting this at this conductivity, I'm getting this concentration. So if I'm getting this conductivity and I know what the concentration is for something around it, then I'll have the concentration. And that's how a lot of our measurements, not a lot, because then we also have a lot of like spectrometers. We'll do a lot of testing, like for COD, that's measuring the easiest way to say this. We have like BOD, COD. That what they actually do is like give us a concentration of the organics inside of the water, the carbon specifically, or not just the carbon, because the bacteria. See, it's hard not to just get. All right, so basically, there's another test that we can do is where we'll take the water sample, we'll have it react with something, and then measure how much light passes through the water at a certain wavelength, so that way we'll know, like, because every different different compounds will reflect light differently, so we kind of have to have a certain wavelength for whatever it is we're measuring. And then kind of like the other machine where I'm using wavelength versus concentration, I do pretty much where I was doing conductivity versus concentration, I'm doing wavelength versus concentration. And then there's also, well, this is the last one, but there's more, but TSS and VSS, because that's also one that we use a lot, which is total suspended solids and volatile suspended solids. And that's basically telling me how much particles are in the water and how much of those particles were organic by pretty much running water through a filter. First, I'll measure the filter, then I'll run water through it, measure or remove the water that was on there by putting it in an oven so that all the water evaporates and all I have is this filter with the solids. So then that's how I get the first part. And then I'll weigh it to find the difference in weight. Then I'll put it in an even hotter, well, we call it a furnace because it's even hotter, I guess. And that'll burn off a lot of the organics that were there. So now I just have all the stuff that's inert and that didn't burn. And then I'll measure that and then kind of have the 3 weights. So that's some of the water testing that I do.
So I think what's one of the take-home messages here is that a lot of the research it's requires a lot of laboratory work, a lot of measurements, a lot of the water quality analysis that we do in the lab requires us to know not only the engineering but also the biology and the chemistry. So there was a question from Sammy. How far have you got in your research? So maybe Daniel, you can talk first, and then Faith.
How far have I got in my research? So I got a long way to go. I came in and I was working on that habits program at first, and that program was just finishing up. Me, I probably have like another 3 years of research, hopefully just 3 years, I don't know. And I mean, not just hopefully, I mean, I would like to continue, but I mean, until I have my PhD, which I'm assuming is part of the question. How far have you got? Oh no, have I got? So right now I'm trying to figure out in those nitrification reactors, I'm using media that has different amounts of oyster shells, and I'm seeing how well the bacteria, the nitrifying bacteria that I'm interested in, how well they're attaching to the oyster shells. If that's giving it an advantage, the surface of the oyster shells, that they're good for attachment, or if it's just the alkalinity that the oyster shells are adding to the water, which is alkalinity helps control the pH, and that's one of the things that we're worried about with these fires. So I'm in the beginning. I haven't gotten very far, but I'm working.
How about you, Faith? You're still an undergrad.
So I'm still an undergrad. So this was an undergraduate research project that I was able to do like as part of being at USF. And so this project technically is over. The PhD student though, who I worked with, he is actually doing his, he's continuing the research, and he is giving his proposal next year. So he's going to be, right now so we found students, the data from students from last semester. And so now he's going to take the students from the spring semester and analyze their data as well and kind of keep the project going. As for me, I'm planning on starting my own research project when I start graduate school in the fall. I'm unsure exactly what that's going to be. Everything's been kind of like crazy with all the quarantine and everything, but me and my professor are going to kind of meet up over summer and get some ideas together for projects for my thesis for master's.
Any more questions from the group? You can always unmute yourself and ask a question. You don't have to put in the chat.
I have a question. So are these research projects like mandatory?
Oh, for me, for undergrad?
Yeah.
Oh, okay. So, actually, so they're not. So pretty much what you do is if you're interested in a certain like type of like research or what professor is doing. For example, I was interested in bioeducation. I went to Dr. Provost and I kind of told her about myself, my interest, and she said that they're kind of always looking for kind of like undergrads for kind of like unpaid help, and so she's looking for volunteers for her lab, and so she had an opening. So I was able to volunteer in her lab and help code Chris's data. From then, I decided to get when we were applying. So if you're volunteering in the lab, you can ask the professor if you want to kind of get credit for the lab. And so I got credit for the lab for this spring. And so as part of getting credit, it's kind of required that you make your own research project and you present it at the undergraduate research symposium in the fall. So the research symposium was canceled with Corona, but we did still finish our research and then kind of do like a little virtual video of it. So it's not required, but if you want to get research for if you want to get credit for your research for graduating, then you will do like a research project in the lab.
Yeah, that's a great question. Also, Daniel talked about his undergraduate research with the algae. And I also, when I was an undergraduate, I never thought I would go to graduate school, but I got involved with a project where we were measuring mercury concentrations in the sediment in Clear Lake in California. And I was out on a boat for days at a time. We were with divers who were collecting sediment samples and camping out with a whole group of people, and it was just so much fun. And so that was really inspired me, and that was part of my motivation for going to a graduate school, that actually getting involved in a research project as an undergraduate.
Yeah, I definitely recommend it. Oh, sorry, go ahead, Daniel.
No, I was like, because like the same thing with me, where I wanted to go to graduate school, but I didn't really picture it until like I got into research, and that inspired it.
Yep.
When in graduate school, most people do research. As an undergraduate, it's more voluntary.
Mm-hmm.
I definitely recommend it. It's definitely been a good experience. I interned at the Florida Aquarium, so I thought I was kind of planning on becoming just kind of marine biologist my first year, in my first year as an undergraduate. But then I did the learning assistant, joined the lab, and kind of really fell in love with teaching and kind of learning more about that. So I definitely recommend just kind of volunteering. And if you're interested in different types of engineering or biology, just kind of there's kind of a lab for pretty much anything you can think of. I would definitely look at kind of the research that's being done at your college, and definitely just kind of send the professor an email or go in person and kind of ask if you can volunteer and get some more experience. And even if you end up not liking it, you now know, you're like, okay, this is something that I don't want to do. But if you do, there you go, you're kind of set. So definitely recommend it. You don't ask, I'll get. So definitely try and get out there and just kind of volunteer and see what you like.
And I've even had a couple of high school students volunteer in my lab. Actually, there's a guy from Middleton who might be working in my lab this summer. His name is Ohas Kalia. And I met him actually when I was a judge at the science fair. And then he asked me if he could come and do some research with us. Any more questions for Faith or Daniel? Okay, so then if there aren't any more questions, we can end the session. And thanks for joining us, everyone.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Bye.
Bye.
I'm gonna stop recording.